Excellent. All right. So thank you very much, Alison and colleagues, for the very nice introduction. My name is Eynon, and I'll be speaking today about exercise, how exercise attenuates epigenetic aging, and I will explain during my talk what it means. I am live and breathe now at the Institute for Health and Sport in Victoria University. This is a beautiful place that I arrived in about 11 years ago and doing my research over here in Melbourne. And my group or my lab is working in the intersection between in the last 5 years, I guess, in the intersection between epigenetics, aging, and exercise. We are also very, very interested in sex differences in physiology and epigenetics. And we have unique exercise cohorts and muscle sample biobank Prof's lifespan in our lab. And we also have access to more samples via collaborators, and I'll speak about it in a minute. I just need to know that you hear me. Does someone tell me that you guys can hear me? Okay,
yes,
great.
We can
Sorry.
Hear you.
Yeah, the interactive thing is sometimes it's tricky. All right, so that's my group. That's what we're doing. And I'll be speaking today about aging and epigenetics. So we all know that aging is a growing health and economic burden on the society. And I can tell you that slowing down aging has significant health and also economic ramifications, more than even trying to solve each disease at a time. Because most of those diseases are associated with age, and we saw that beautifully in COVID recently. We know that aging is accompanied by decline in functional capacity across the lifespan. And in fact, across every organ system, age stands out as the single biggest driver of disease. And as you can see here, all individuals will experience changes with chronological aging, but some individuals will have different biological age. And what it means is that their functionality will decrease earlier in life, and they will lose their health span. And health span, and this is what we need to focus on, is the number of years that we live in good health. So instead of just elongating life of people, which is accompanied by health span, we need to make sure that they spend in the 10 or 15 last year of their lives in good health. So this is the sort of the entire mission of the aging research. And as you can see here, some people will, have their biological aging plateau for many, many years in a good health, while some adults are declining very quickly. And one of the hallmarks of aging is epigenetics. So what is epigenetics? Epigenetics are simply modifications in our genome that affect the expression of the gene. So these are modifications that, as a consequence, the gene expression is changing. And epigenetic changes stand out as one of the hallmarks, the big hallmarks of aging. This is a recent paper, not a recent, but 8 years ago, a big review paper in Cell showing that epigenetic, along with other modifications, is very important for aging. And within the epigenetic, DNA methylation is the most studies and the hallmark that we'll speak about today. So what is DNA methylation? DNA methylation refers to an addition of methyl tag to the 5th carbon of a cytosine residue on the genome that reside alongside the guanine base. And we call them CPG sites over here. And those CPG sites are distributed across the genome in many, many places, like enhancers, for instance. They are very dynamic, so they can change exactly like gene expression. And Can you hear me now? Sorry.
Yes, you muted yourself.
Yeah, I muted myself. Okay, sorry. Okay. So the methylome, as I said, is dynamic, and CpG sites can be added and removed by DNA methylation enzymes. And many CpG sites has been shown to change with age. So as you can see here, this is birth and age, and you can see this is a specific CpG site across the genome, and it can be unmethylated when we're young and then get methylated across the genome when we're getting old. The interesting thing is that we are looking at that, or we measuring CpG sites on a gradient, although it's a binary thing, either methylated or not. We are measuring that on a gradient of 0 to 100% across the genome, across the cell population. And there are two ways to assess epigenetic aging. One of them is the epigenetic clock. You might have heard of that, and if not, I will elaborate in a minute. And the other one is epigenetic wide association studies, exactly like GWAS, genome-wide association studies. So, epigenetic clock. If you haven't heard about this concept, this is Steve Horvath, Professor Steve Horvath, our collaborator, and he published the first paper in 2013 showing that there is a highly accurate biomedical test that is called epigenetic aging that can be used to measure age based on DNA methylation patterns or specific DNA methylation patterns across the genomes. So specific CpG sites, according to this algorithm, can predict our chronological age. And interestingly, it's multi-tissue. So when he published that in 2013, he showed very beautifully that it's across many, many tissues in the human body. It's predicting age. Very accurate with R that is more than 0.8 across tissues, which is really accurate. It's a good predictor of age across most tissues, and he also published a lot of studies in other species showing the same pattern. So very well replicated. And the differences between the estimated DNA methylation on the right-hand side and chronological age might be biologically meaningful. So it's called age acceleration. You have positive epigenetic age acceleration. Let's see over here. You also have a negative epigenetic age acceleration, which is probably better. Now, the only tissue that Steve haven't really accurately looked at is skeletal muscle, just because he didn't have skeletal muscle samples back then. So what he did with his algorithm that he developed, he tried to predict as much possible what would happen in skeletal muscle, but his prediction was not very good. In fact, it was very poorly prediction in skeletal muscle. So because we are very keen in my group on skeletal muscle in humans, we are very specialized on that. We embarked on this mission and created an epigenetic clock in human skeletal muscle. So this is Sarah Voisin from my lab. She created the first epigenetic clock for human skeletal muscle, published in 2020 in JCSM, and called or named by the convenient, beautiful name MIT, Muscle Epigenetic Age Test. And on the left side, you can see that this is the first MIT clock based on 682 samples, which is quite a lot in muscle. We're able to data mine, when I say we, I mean Sarah, we're able to data mine more, close to 700 samples from 12 data sets with epigenetic data, and with an average correlation on 0.6 and a median error of 4.6 years. And those CpG sites in the clock that were the most predictive of age were around 200 CpG sites. When we got more and more samples during the years or during the last year, we accumulated that. So it's an ongoing process, and we created MIT2, which is more than 1,000 samples. And we sort of made the correlation a little bit more accurate and more, and you can see it on the right-hand side that the median error has dropped. not significantly, but it's dropped. So the more we add samples, the more we see the accuracy of the mid clock. So again, it's a good predictor of chronological age. Now we seated and waited for replication, which is one of the most important, I guess, steps in science. We were very anxious, to be honest, to see that it's working. And just before that, I want to mention that we put the mid clock, or Sarah put the mid clock in bioconductor as a package, muscle epigenetic age test, so other people can use that, and it's freely can be freely used by any scientist in the world. So yes, we got a really nice replication study from people in Norway, published in Clinical Epigenetics. You can see that the R is very, very highly correlated, the age, with our DNA meat test with their samples. So that was really reassuring for us that it's working. And then another group found a weaker correlation, but still very significant, with the chronological age and our aging clock. So that shows us that this clock is in muscle is working and it's predicting chronological age beautifully. Now, the thing is with these clocks is that those CPG sites, according to the algorithm, are trained to capture methylation sites that are, or this algorithm, sorry, should say, is trained to capture CPG sites across the genome that are changed mostly with age, strongly changing with age. It doesn't necessarily mean that those CPG sites will change with environmental stimuli like exercise. And two papers, recent papers, one in Aging Cell and the other one is the same study in Clinical Epigenetics, actually showed that across 3 different clocks, the Horvath clock, the Grimmage, both of them are clocks that are trained in blood, and the mid clock, which is an epigenetic clock in muscle, our clock, there was no correlation between physical activity and the epigenetic clock. So we're back to square one in terms of environmental factors, in particular physical activity and exercise, on how does it change the DNA methylation. And that also reiterating for us that the clocks are not optimal model to assess DNA methylation changes in response to exercise. So next, what we did is we went to epigenetic wide association study, and epigenetic wide association studies are studies that are looking at CPG sites across the genome that are changed with age, not necessarily within this algorithm, and not necessarily those sites that are only changing with age and highly predictive of age, but also sites that are being changed with exercise and other environmental stimuli. So to answer the following questions, so because we are very interested in exercise, we wanted to answer a few questions. First of all, do fitter individuals, people with higher fitness at baseline or lower fitness with baseline, display younger methylation and transcriptome profile than less fit individual? Now, we also looked at transcriptome. As I said before, the DNA methylation is going side by side with gene expression, and changes in DNA methylation are often very highly correlated with changes in the transcriptome. So we looked at both at baseline. Then we looked at exercise. So we wanted to ask, does exercise training mitigate or reverse omic aging in muscle aging, in human muscles, sorry. So data sets with exercise training. And then we looked at, I guess, the other end of the spectrum, which is immobilization data that we had to look, and which we think should be clearly look like an aging profile or the other spectrum of fitness, high fitness. So we sort of have for us a control in this sense to look at all the end of this, 2 ends of the spectrum. So that's the muscle disuse. So what we typically do in our lab, we're taking muscle biopsies from people. This is the GeneSmart study, gene skeletal muscle adaptive response to training. Currently have more than 150 participants in this study with several data sets and several points that we take in our collecting biopsies. We train those people several weeks, typically between 4 to 12 weeks of high intensity training, taking biopsies, and of course we are collecting all the physiological data before and after, VO2 max, peak power, and a lot of other physiological data. So the first thing that we did before we got into the exercise data, we looked at EWAS, epigenetic wide association study meta-analysis of age in skeletal muscle. So we wanted to look and see whether those CPG sites, whether the methylome is changing because of age, purely because of age. Okay, so we did look at that, and you can see on the right-hand side or on the red, you can see that CPG sites or DNA methylation sites, the change or increase the methylations, hypermethylated, just because of age. And on the left side, you can see in blue, you can see the sites that are reducing their methylation just because of age. And it's quite widespread. More than 40,000 DMPs, differentially methylated positions of CPG sites, are changed just because of age. So that's the first step for us to know that, you know, the methylome is changing because of age. Let's, in this specific method, let's see what's happening with the exercise. We also looked at transcriptome and proteome with other data sets and tried to correlate and integrate those data together. And we found 57 genes that are common between all those omic layers. This paper was published last year in JCSM. That's the first step before we're looking at exercise. We also created a website called MetaMeth or metamethylation, which you can actually look at your gene of interest and see how it changed with a gene or gene of interest or protein of interest, and how the integration between omics goes. So I encourage you to go and find this. Just press on Google MetaMeth and you will find it and see how it works. So it's really nice, freely available again, and we strongly believe in open science. So we are, you know, fostering the open science and trying to have everyone look at this website and our work as well. All right, let's come back to the exercise. And the first step, as I remind you, was the baseline. So does regular exercise or long life exercise is associated with DNA methylation aging in human skeletal muscle via the EWAS. So the first step was to identify omic data sets with physical activity or fitness phenotypes, and then to look at data sets that are following exercise training or inactivity, which is not easy to find human muscle samples in all omic layers. We looked at cross-sectional and then longitudinal interventions. We use open access repositories and our own lab, the GeneSmart, and our collaborators. So this was quite a big undertaking by Sarah Voisin, big data mining. DNA methylation and mRNA, you can see the data sets that we dipped into. And across the data sets, we have more than 500 DNA methylation data sets, samples, and almost 1,500 transcriptome, which is more, more studied in the last few years. And this is the data sets that we looked at. Yeah, so we performed meta-analysis in DNA methylation and transcriptome, and we didn't look at proteomics just because we didn't have enough data. So if anyone in the crowd or anyone that you know have proteomic data on exercise in skeletal muscle, please contact us. We're very keen and very interested with that. We have some data on that, which has been bioarchived recently, and we'll have another paper on proteomics and exercise soon. But we need the big numbers. So the next step was to test the association between age-related CPG sites, mRNA, and fitness exercise or inactivity in each individual study. Then we meta-analyzed everything to see if those CPG sites with aging are different or changing differently than the one that are changing with fitness. And I will try to explain that in a minute. This is an unpublished study and hopefully will be published early next year. So, results. Higher aerobic fitness is associated with younger omic profile. On the left-hand side, you can see the methylome. And you can see a clear negative correlation between DNA methylation changes with age and DNA methylation patterns that are changed with exercise. And I will try to explain that. On the y-axis, you can see that VO2 max, and on the x, you can see the age. These are CPG sites in the transcriptome, in the methylome, and genes in the transcriptome. And those CPG sites, these are CPG sites that are reduced with age, reduce their methylation or gene expression with age, and increase their VO2 max. Okay, so negative correlation between them. And as you can see here, we see a beautiful negative correlation in both omic layers with an FDR of 0.05 in both methylome and transcriptome. And at some CPG sites, and that's really interesting, and transcript affected by both age and VO2 max, we see that every increase of, you can see here, every increase of 1 milliliter minute kilogram in VO2 max is can be explained by 2.9 or 1.9 years. Okay, so every increase in 1 unit of VO2 equivalent to reduce of 2 or 3 years of age. But this is, of course, only one or specific sites in the methylome, and I should state that it doesn't happen in the whole genome and only in some CPG sites. But it's remarkable and amazing that there are CPG sites and DNA methylation sites that can actually increase, with the increase of VO2 max, can shift their pattern in a way that you look almost 2 or 3 years younger. So I hope I explained it properly, because it's a bit of it's a bit of hard thing to explain, but also to understand, just because it's heavily statistical. We used 3 independent cohorts in the methylome and 4 independent cohorts in the transcriptome, 350 to 400 participants, which is quite good in muscle. When we looked at exercise training, does it mitigate the aging, the omic aging? Then we weren't very statistically robustly powered in the methylome. We did see very nice trend. Okay, you can see it here. It wasn't quite significant. In the transcriptome, because we had almost triple of the sample, we showed significant reduction or negative association between, again, exercise and CPG size that are changed with age. So they kind of negate each other. Then we looked at muscle disuse. Does it accelerate transcriptome and methylome? Now, unfortunately, we don't have, and I'm not aware of any data set with this model in the methylome, but we did have access to one data set in the transcriptome, and we show exactly the opposite. So what happened with aging is that you can see here, when we looked at the muscle disuse, we see that it's actually promote aging. And physiologically is the opposite from exercise. And the DNA methylation, we showed a positive correlation between aging, related CPG sites, and inactivity, going in the same direction of aging. So hopefully I made myself clear here. So maybe this one is a good way to show that as well, or even a better way. So if I summarize the data, you can see that each row here, sorry, just going to try here. Okay, each row here is a CPG site or a site across the genome. And in blue, we have CPG sites that decreased the methylation with age or muscle disuse. Whereas in red, you can see that CPG sites that increase with fitness or following exercise. So again, contradicting each other. You can see here, CPG site, decrease with age, increase with VO2 max, but also increase after training. And as you can see in the transcriptome, beautifully, you can see that age and muscle disuse are going in the same direction in those DNA methylation sites, whereas exercise or higher VO2 max at baseline and training are going in a different direction. And it's really nicely across the human methylome and transcriptome as well. All right, so these cohorts are, as I said, difficult to get. So if anyone has human cohorts with biopsies and exercise in South Africa, we'll be very happy to collaborate and very happy to hear from you. We are now performing integration to pinpoint the top genes that are related to those changes in the methylome and transcriptome, because ultimately we want to pinpoint the genes that are most predictive and most important in these processes, and then target them and try to do some functional work with using vector to methylate the promoter of genes and also sRNA. And one of our difficulties we have is that we are not aware of cell culture model that can mimic long-term exercise. We are aware of cell culture models that are, very short and that can mimic, I guess, short bout of training, but not long-term exercise. So this is the difficulties that we have with human studies. And as you can see here, the omic integration and the CPG sites, again, age and inactivity are going in the same direction, both at the methylome and the transcriptome. Yeah, so again, the functional validation of top genes, this is what we're looking at, the unmethylated promoter versus the methylated promoter to see if those genes are really predictive of aging. So that's the future of what we are doing at the moment. I want to touch on one last point that is related to other DNA methylation patterns in the human genome. So those DNA methylation patterns that are spoken about it today, called DMP, differentially methylated positions. And they're the one that everyone is looking at. But in fact, there are many, many other not many others, but some other DNA methylation patterns across the genome that we hypothesize that are more predictive of biological age. So this is Kirsten Seal, a former student of Alison. I'm sure that Alison now is smiling in the crowd over there. She's one of my best students, and I'm very thankful to Alison and South Africa for raising her and brought her to Australia to work with me. She's very good in bioinformatics, and she published this year a wonderful paper in Nature Reviews Genetics, and there are more papers to come in big journals, which I'm hoping to be early next year. So Kirsten is looking at what we call VMPs, viable methylated positions. And without getting to too much explanations, those sites in the methylome are more chaotic. So they're not changing in the same directions like DMPs during aging. They are being more, they capture differences in DNA methylation at older ages. They are being more, I guess, chaotic is a good word, and more viable. And they change the viability with age. So Kirsten is hypothesizing that those changes are more important biologically, and she's currently undertaking a big study with more than 50,000 samples across tissues to look at those VMPs across all human tissues and to try to integrate and to pinpoint those VMPs that are, I guess, tissue specific, but also shared between tissues. Okay, so if I'll summarize my talk today, so age has profound effect on skeletal muscle epigenome. Show that and publish that. Individuals with higher fitness at baseline have younger DNA methylation and transcriptome profile. Again, we show that across many human samples. Exercise training shift DNA methylation profile. It wasn't significant, but it does shift. We just need more samples, but it definitely shifts the transcriptome towards that of a younger profile. We also identified some of the top genes associated with the interaction between aging and exercise in muscle, which I didn't show, I didn't specify them today, but we do have them. We are now validating some of those genes using methylation vectors and siRNA just to make sure that we have causality over there. And those genes are really predictive or changing with aging and are important to exercise. So with that, I would like to thank my lab, in particular Sarah Voisin, who's this is mainly her work, and the other members of my growing lab. Also collaborators from across the world, Steve Hovat, Andrew Teschendorf, who is a highly regarded bioinformatician, and other people that contribute to this phenomenal work. Also, the Australian government and the Australian Research Council, ARC and NHMRC, which are funding my studies and the study of my wonderful book. Thank you so much.